I'm Daniel Levine, and this is the Bio Report. Human leukocyte antigen, or HLA genes, help the immune system tell the difference between the body's own tissues and outside threats. In some people, certain versions of HLA genes mistakenly flag normal proteins as dangerous, which can push the immune cells to attack joints, nerves, the gut, or other organs. Most autoimmune diseases are driven by changes in HLA genes. RheumaGen is developing a new kind of gene editing treatment that aims to cure autoimmune diseases by going after one of their root genetic triggers. Instead of broadly weakening the immune system, the company's goal is to switch off a single bad version of an immune gene while leaving the rest of the body's defenses intact. We spoke to Richard Freed, CEO of RheumaGen, about the role of HLA genes in autoimmune diseases, how the company's gene editing therapies work, and its lead program in rheumatoid arthritis. Richard, thanks for joining us.
No, it's a pleasure. Thanks for having me.
We're going to talk about human leukocyte antigens, or HLA, their role in autoimmune diseases, and RheumaGen's efforts to develop HLA gene editing therapies designed to cure these conditions. Let's start with human leukocyte antigens. Can you explain what these are and the role they play in the immune system?
So at RheumaGen, we are editing the HLA gene. So the HLA gene, or the immune gene, codes for these proteins that sit on the surface of all of our cells. And their job, these HLA proteins, their job is to present peptides to T cells to start the immune response. Usually it's viruses or something foreign that they're saying this doesn't belong here. Let's show this to T cells. There are tens of thousands of different HLA alleles out there, or flavors, right? Each of us has about a dozen, which makes up our own, unique flavor profile, if you will. And normally, when they present viruses to start the T cell activation, that's what you're looking for. But when you have an autoimmune disease, they mistakenly start presenting something of your own as foreign. For example, in the case of rheumatoid arthritis, it's the collagen in your joints, or in MS, one of them is the myelin in your nerve sheaths, right? It's been very well known for many years that certain HLA types are highly linked to certain autoimmune diseases, and others are highly protective. But nobody really knew why. And that's kind of how that was sort of the beginning of this research was digging in a little bit deeper of what makes you susceptible versus protected to different autoimmune diseases.
What role do they play in autoimmune diseases? Is there some dysfunction or problem with HLA genes that underlie these conditions?
Yeah, so of the tens of thousands of alleles out there, there's only really, call it a few dozen HLA alleles that tend to be linked to the majority of autoimmune diseases. And there's lots of different ways that it can happen. Sometimes in RA, for example, you can have a lung infection, and that particular antigen just happens to look very similar to one of your self-antigens, and then that particular HLA allele starts to mistake in that, and then it just starts presenting, and the more and more antigen presentation that you have, the harder it is to get the disease back under control.
What problem were you trying to solve when you started RheumaGen, and what convinced you that HLA editing could be a way to not just treat but cure autoimmune diseases?
So as I mentioned, it's been well established that certain alleles are highly linked to these, but wanted to take a look at what made that the case. So Dr. Fried and his team, they looked at the crystalline structure of all these HLAs, and they noticed that there's one amino acid difference between someone that is highly protective and someone that's highly susceptible. But here we are, 10 years after the advent of CRISPR. What? Why has no one else tried to edit HLA for some kind of therapeutic benefit? Seems kind of an obvious target, right? But that's because it goes against probably about 40 years of immunology dogma in saying that any mismatch to HLA gets rejected. So, in the same reason, there's about a 1 in a million chance that I can give you my kidney that we have the same flavor profile that matches up. The thought of, well, if you make any changes to that, it's going to be rejected because it always is. But Dr. Fried and his team, where this came from, the University of Colorado, they're HLA geneticists, right? They look at this all day long, and they are the ones that are responsible for doing the matching for organ transplants that happen across the different hospitals in the state of Colorado. And they noticed that in the, HLA looks kind of like a taco or a pita pocket. And the one area that was different, the areas that we were thinking of editing to modulate what actually can be presented in there, is way deep down in the bottom of the taco, right? And there's stuff that that sits on top of it, and the T cell comes and tries to look at the whole thing. The idea was, well, the change that we're making is so deep down buried into the HLA groove that the T cell won't notice the difference. And that was the bet the company experiment a few years back, where we put human HLA into mice and did skin transplants between them, and lo and behold, you can get away with that. And why HLA is so exciting is because the HLA T cell interaction point is really the start of the disease interactions, right? That's the factor. That's where it all begins. We're going and changing things at the source. So if all other therapies allow that HLA T cell interaction point to happen, and then try to put the genie back in the bottle of cleaning up the inflammation downstream. So HLA was a very tantalizing target, but no one really looked at it because it seemed to be impossible.
Well, let's talk about RheumaGen's HLA gene editing platform. What are the common elements that run across all of your programs?
Sure. When we started this, we were looking at in each of the different indications, we were doing it a similar but slightly different way, right? So essentially turning you from the highly affected phenotype to the one that's highly resistant. And we collect the cells the same way. Everything would be done the same, but each one would be slightly different. So we would have to have different clinical trials for each. And that's great when you have autoimmune disease where the antigens are really well established, right? For example, in celiac, it's very well known that it's the wheat, gliadin that is the problem, and that alone. But in a lot of autoimmune diseases, it can be that naughty HLA allele that's acting up can be presenting a couple of different things, right? So in RA, there's the collagen in your joints, but there's also these citrullinated peptides. There's a few different things. So when we're trying to make these very specific point mutations to turn off one antigen and one antigen only, sometimes that would only solve part of the problem for these patients. So as we, kept evolving this over 5 years, we developed the concept of what we're calling anchor editing. So rather than try to find the differences between HLA alleles and what makes someone different from someone else, we were looking at, all right, what's kind of the same? What is fundamental to HLA presentation in general? So of all of the polymorphism of HLA, all the tens of thousands of alleles out there, all of us have asparagine in position 82. So the reason for that is when it binds to peptides, and peptides, there's lots of different polymorphisms of peptides right out there, but all peptides in this spot on the spine of the peptide, if you will, is always the same because it has to link up to the HLA presenting asparagine. So basically, by swapping a different amino acid in there that doesn't have that ability to do the hydrogen bond, we essentially kind of turn off that HLA allele altogether. So maybe turn down is a better way to say it. So it's not going to be presenting anything that it was presenting before, or if it does, it's going to be much weaker. But you still have all of your other 11 or 12 HLA alleles that provide lots of cross coverage that you can still mount a very strong immune response to anything. Probably won't really even notice much of a difference if you just knock out the one, because we're just taking out that one that is essentially now really most of what it's doing is presenting self antigens. So by taking that one out, we think we can knock everything down. But the beauty of this also is that having the same target now, so now we've pivoted now to go after this anchor editing target, which is the same across all of our indications. So that helps us to take advantage of the platform designation, for example, from the FDA, with the idea of once we show that it's safe to edit this position 82, this anchor edit in humans in general, then we can skip ahead to phase 2 in the other indications, right? Just focusing mostly on the efficacy.
How does your anchor position editing strategy differ from prior gene editing approaches that attempted to correct or knock out disease associated alleles one by one for each patient subgroup?
Yeah, this one cuts across all of the class 2 HLA alleles. So rather than trying to figure out exactly what is the antigen that HLA allele is presenting, in some cases we don't even really care, right? Whatever it was presenting before, it's going to stop doing it now. So with this approach, it's much more universal. It cuts across multiple indications, and it also opens up the possibility that we could do this in rare autoimmune disease as well. We've started going after these larger indications, but now since we're doing the same edit, it's much easier to start a program if someone has an interest in a particular area. They know what HLA allele is linked to it, then just turn that one off as well. So it's we can provide the kind of the economies of scale of the large indications that into those rare diseases.
Walk us through the process of how you go from the patient to the treatment, and what are the biggest scientific or manufacturing challenges in that?
And this is an area that we've been very intentional over the last several years of trying to simplify the gene editing process as much as possible. Right. We it starts with the collection of hematopoietic stem cells from the patient. That's done clinically all over the world. That's just what our lab here at the University of Colorado supports lots of these. So we collect these hematopoietic stem cells, and first we're going to be shipping it here to Colorado through our lab to do the gene editings. And then we were able to essentially just make that one change to turn off the body's ability to continue attacking itself. Then we can freeze down these stem cells and ship it anywhere in the world where it's infused back into the patient with a one-time outpatient therapy. So essentially, hang the bag, thaw it at the bedside, and we can do this in a way that does not require myeloablation, and that's I think a big step for the field. In that, in the past, the cell and gene therapies, in order to get the stem cells in, they use kind of a sledgehammer, right? So it's been what has been used over the last 30, 40 years in hematopoietic stem cell transplants for leukemia patients and other blood cancers, right? So they have to, it's kind of a big sledgehammer because you have to kill cancer cells. You have to kind of wipe out the immune system. It's a much bigger thing that they're trying to accomplish. And a side effect of that is it opens up space in the bone marrow, right? So all we need is to open up a little bit of space in the bone marrow for our cells to engraft. So we don't have to use the much larger doses that have been used in the past. So we think that we can do this in a way that can be done in an outpatient facility, in a hospital adjacent type facility, to start infusion center, kind of where RA patients and other patients get their typical medication infusions.
What's known about the preciseness of the therapy, and have you seen any evidence of off-target effects?
So we are going through our IND enabling studies now that will be putting a much clearer picture on all of this and all the off target effects. But our initial studies so far have shown no off target so far. And part of the beauty of the polymorphism of HLA of how many different alleles out there, it's somewhat difficult to have an off target effect, much more so than in some of these a gene system that isn't quite as different.
Your lead experimental therapy is RG0401, an experimental therapy for the autoimmune condition rheumatoid arthritis. Why start there?
Starting in RA, it has a very strong HLA association, and it's a very large indication. About 1% of the global population has it. And there's a significant proportion of RA patients who don't respond well to current medications, right? So there's been uh, a lot of advances over the years in RA and other autoimmune indications with the Humira and other biologics, but 60% of patients still never reach remission, and about 20 to 30% are known as the refractory patients who have failed multiple different medications and are now on the path of 15 years of failing drug after drug after drug. So it's an expensive, difficult way to treat the disease. So it made a lot of sense to start there, and but now we think we can move into other autoimmune indications as well.
How is the condition generally treated today, and how effective are existing medicines?
So it's treated with a variety of things. It's starts with like a methotrexate, like a disease modifying agent that's been around for a long time. Most patients fail that and then are added a biologic like a Humira or different inhibitors. So essentially, what they're trying to do is once T cells have been activated, T cells then talk to B cells, and then they call up macrophages, and there's a lot of immune signaling pathways downstream. So that's really a lot of it is trying to deplete that. You're trying to gum up the works and trying to slow down the process. And then there's some others that do depletion of B cells or things of that nature. They are there have some benefits that just it's certainly better than not being on medication, but they tend not to be curative. And patients, about 75% of patients are unsatisfied with their therapies. So we think there is a it's a doctors aren't happy, patients aren't happy, and insurance companies and the payers aren't happy. So we think this market is ripe for disruption with something that can take a step change improvement on the current therapies.
You spoke in general terms about the platform, but in the case of RG0401, what does it do at the DNA level, and how does this change antigen presentation?
RGO401 is our lead indication. So we're going after the DRB0401 allele. So it's one of the most prominent alleles in rheumatoid arthritis. There's about three or four that make up most of the vast majority of patients have three or four of these different alleles. And essentially, what it is going to be is going to be a gene edited cell therapy. So essentially, we're collecting those cells, editing, basically turning off the body's ability to keep attacking itself, infusing the stem cells back into the patient, where then they migrate to the bone marrow, and then they start to produce immune cells that can no longer trigger the autoimmune response, right? So it's essentially where think of it as changing the factory settings in the bone marrow to then have your hematopoietic stem cells produce immune cells that can present lots of different things, but they can no longer present the collagen or the whatever in your particular autoimmune disease is going on. So then within a matter of a few days, your old cells get replaced, right? So every day you get new antigen presenting cells that come out of the bone marrow. They live a few days. But so as this therapy goes for a few days or weeks, you should have most of your antigen presenting cells in the bloodstream have been replaced. Then after a couple of weeks into the tissues get replaced. So we should be able to see these patients having significant benefits and feeling a lot better within a couple of weeks. And another important thing to think about is we don't actually believe we have to replace 100% of their stem cells, right? And it's unlike oncology, where if you get 95% of the cancer cells, you're going to have 295% of the problem 6 months later, right? In autoimmune, it's different. They're slower and develops more slowly and chronically. So if you get 80, 90% of the cell replacement, the patient's probably going to feel 90% better. And that might even be a tipping point to fully cure the patient where they can get off of medication in general. So a low T cell signal actually can be what's known as tolerogenic. So it's something kind of like the boy that cried wolf, right? So if they see like a weak signal, then the T cells just kind of ignore it. So we think we can get to that point where we can cross the tipping point.
What's known about it from the studies you've conducted to date?
Well, we've shown that we can block 100% of antigen presentation of all the major self antigens that have been studied in rheumatoid arthritis and multiple sclerosis, and type 1 diabetes, and celiac. So we've shown that not only the antigens aren't presented, then T cells are also not stimulated. We've taken that into the mouse models. So in rheumatoid arthritis, so we've shown that our mouse won't develop RA. So once essentially you give a few injections of collagen in the tail of mice, and they start to develop what's called collagen-induced arthritis. But with our mice, they don't develop it, and then we do a bone marrow transplant between the mice and show that that protection can be transferred from mouse to mouse. So that's been exciting data. We'll have similar data in MS and T1D coming up this year. But then ultimately is to try in patients, right? So there's no non-human primate model that is necessary for this. This is kind of come just about as far as we can go in mice, and now it's actually time to try in patients.
And what's known about the durability of the therapy at this point?
Well, we suspect that it'll be permanent, right? Because a lot's been known about hematopoietic stem cell transplant over the years, right? So there's lots of data, for example, in pediatric transplants of the graft still being there 40, 50 years later. It's a little hard to get data past 10 years in adults, because these are typically done in late stage leukemia patients that tend not to live that long, but their stem cells are doing fine. But it's so as long as you are able to edit the long-term progenitor cells, then get enough of those edited, we think this should be a permanent solution.
What's the development path forward? Have you settled on a patient selection, endpoints, or biomarkers for the study?
Yeah, so we'll be collecting cells from patients in Australia. We are going to be going for rheumatoid arthritis patients, so it's not going to be healthy volunteers. It'll be really 9 to 12 RA patients who have failed multiple biologics, so they meet the definition of refractory. And while it is a safety trial, we are primarily focusing this as a safety trial for the platform in general, right? But since we are going to be treating RA patients, there's going to be a fair amount of efficacy signals that we're going to be looking for, right? To see if they've, reduced their inflammation, or are they able to come off their meds, and a variety of other scores as well. So it will be indicative, but then that'll help us to then, raise the Series B to then go into the larger clinical trials with both RA, and then we perhaps moving on, in MS as well.
A lot of companies have been hesitant to use the word cure. RheumaGen uses it prominently. Clinically, what gives you comfort with that language in RA?
Yeah, that's a good point. We do lean into it. We're not interested in incremental improvements, and this comes from the biology of the diseases that we're treating. For example, we it's well known that these diseases are HLA T cell mediated, right? So if we're the only ones that can impact what's going on at the source of the disease. That's why we think we can treat these untreatable patients and get them to a point where they can be cured. Uh, so there's the biology of it, but then there's also, it's been shown in the past, patients who have leukemia or AML or something, and they get a bone marrow transplant, but they also have RA, or they have an autoimmune disease that they're cured of the autoimmune disease because of the different HLA. It's a little bit of an HLA mismatch when you have the bone marrow transplant, right? So it's not quite a perfect match. So they match it just enough, so then they're able to not uh, continue presenting the peptides, right? But that's always a delicate balance because you have, in traditional allogeneic transplants, you have to be on lifetime immunosuppressive drugs, right? So it's that balance of curing, stopping the disease, but it's a much more serious and dangerous, I guess, procedure that we typically wouldn't see for just the rheumatoid arthritis patients. But what we're doing is essentially combining the safety of an auto transplant, so the body doesn't notice any difference between our cells, the edited cells, and your own cells. So we can so it's a much milder approach, right? We don't have to worry about the rejection, right?
What do you know about how regulators are thinking about a one-time HLA editing therapy for a common autoimmune disease compared with how they look at rare disease gene therapies we've seen approved to date?
It's a good question. And that was one of our, I think, biggest concerns going into our discussions with the FDA a couple years back when we had our Interact process. Was no one had done gene editing in a larger disease or something that would be, while rheumatoid arthritis is severe and it's generally not considered fatal. But there was no hesitation at all. They seemed very excited by the prospect of what we're doing. There was no concern of the ha- of a larger indication as opposed to what has been done in the past. And frankly, I think we owe a lot to the pioneers in this field that came before us. Right, they went after these rare indications that if we were trying to come in first with RA, a big indication, that would probably raise some eyebrows. But the fact that they showed to be safe and successful, gene editing in general, that really opened the doors for us. And that was part of our strategy, right? We bet that by the time we're ready to be in front of them, they'll be more comfortable with this, and we can go after a disease that is much more commercially viable or commercially exciting, even in rheumatoid arthritis, as opposed to some of these rare diseases where it can be hard to make the economics work.
There are some concerns about the cost of cell and gene therapies. Given the indications you're pursuing are large markets and not generally life threatening, how are you thinking about things like cost of goods and manufacturing and ultimately pricing and access?
Yeah, we've said from day one that this therapy can't just have an exciting clinical value proposition, right? We want to treat millions of patients someday. This needs to be not only commercially viable, it needs to be commercially exciting, right? So from the beginning, we wanted to keep this out of hospitals, keep this away from that. You've seen these $1 million, $2 million price tags. Uh, so we have been able to engineer this so it is considerably less expensive than that. And now we even have a new approach using a bacterial-based gene editing system that can bring our cost of goods down even dramatically lower, right? So we think we can do this for a payback period of 2, 3, 4 years, because what we're competing against in these autoimmune indications, there's chronic lifetime care, right? Expensive drugs, 50, 60 grand a year just in the medications. not to mention all the surgeries and the comorbidities and the complications and people not being able to go back to work. And so there's a huge economic benefit from what we think we're doing here. And we think that it's going to be exciting for them, right? So they can actually consider this a healthcare investment as opposed to expensive therapy that they have to go find money for, right? So while the while it sounds great sometimes to develop a drug for in a market that has no therapies at all, there's nothing. It's clinically exciting, but commercially, then that payer needs to find money to cover that from somewhere else, right? But if we're replacing existing spend, then that's a whole different ballgame.
And how is the company financed to date? What's the conversation been like with investors?
We started the company in the summer of 2018. We got our first seed round closed right before the pandemic in November of 2019. Is a family office out of Texas took the whole round. And that was kind of in the boom days of cell and gene therapy. And then COVID hit, and then things went really crazy. And watched the market go way up and come back down. But we've been able to sit on the sidelines and ride that biotech wave a little bit better than others because we've got a much lower burn rate and we've got a great partnership model with the university, right? So that's been able to advance this technology for somewhat of a fraction of the traditional spend you think of a biotech company raising hundreds of millions of dollars when the technology is really in the infancy to build labs and to hire scientists that are already in place here. So we just essentially contract back with the University of Colorado, the lab that this came from. They have all the ability to take this all into the clinic for us. So we can do that as a kind of a fraction of the traditional cost. And then now we have raised our Series A round. So last JP Morgan conference, we announced that we closed that, which will take us, it was a $15 million round, which will take us through our first in human trial. So that will start to get our first human data from rheumatoid arthritis patients. But also we hope to move along MS and T1D into the kind of ready to go stage once we show that it's safe in humans to edit this anchor position, then we can kind of move ahead with the other clinical trials as well. So it's been interesting conversations with investors. Everyone's excited about it. It's new, different. No one else is doing this, which is exciting. It's also sometimes it's a little lonely being the only one, but we wouldn't have it any other way.
The most recent financing round included an investment from the nonprofit Beyond Celiac. What role do you see patient organizations playing in advancing novel therapeutic approaches that may not have been de-risked enough to attract pharma and traditional investors?
Yeah, it's been great with Beyond Celiac. So we opened up an additional Series A extension round to advance work in celiac and some of the other indications, but also to look at an in vivo approach to gene editing. So what we've talked about so far has been the ex vivo, with collecting cells from patients and then editing in the lab and shipping it out. But ultimately, the goal is to be able to deliver that gene editing cargo directly into the body, right? Edit the stem cells where they are in the bone marrow or maybe into the bloodstream. Being able to have more of an off-the-shelf approach then we can start to reach the millions of patients out there, right? But with people like Beyond Celiac and others, they help to really get that patient voice into the conversation early, right? To be honest, we were a little bit surprised of the different indications that we have, MS, RA, spondylitis, type 1 diabetes. We kind of viewed celiac as the somewhat less, severe of them. But as they educated us, while celiac itself isn't fatal, people with refractory celiac that can't just avoid gluten, they tend to have lots of stomach problems. colon cancers and those types of things. So it becomes a much more serious disease than you really expect. And it also helps you to always keep the patient's voice in the room and in mind, right? So at the end of the day, is this something that they would be comfortable doing, right? Are they gonna line up? Are they lining up for this, or is this something that takes a lot of convincing? And so far, the outpouring from patients has been almost overwhelming. It's been kind of everywhere we go, we talk about this, and people come up to you and because everyone's got someone that they know, right, that has one of these indications. So it's been great having them on board, and it also helps to provide a bit of validation as well, right? So if pharma and other investors are looking at this, it's a new target, this is very novel. But if these patient organizations come in, then that helps to uh, provide that next level of understanding.
Richard Freed, CEO of RheumaGen. Richard, thanks so much for your time today.
Thank you very much. It's a pleasure.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.